To copy 3.2 billion base pairs with near-perfect accuracy, the cell relies on an exquisitely precise biochemical machine. But DNA replication is a physical process, and like all physical processes, it is susceptible to structural errors. At the molecular level, a mutation is a permanent, heritable, and relatively infrequent alteration in a nucleotide sequence. These alterations are not biological accidents in the traditional sense, but the inevitable consequence of thermodynamic principles acting on molecular bonds. No complex system can replicate without error. To understand how these microscopic changes dictate physiological outcomes, we must classify them through a systematic taxonomy. This framework organizes mutations across 4 primary branches: the genomic side of the change, its translational consequence, its fundamental source, and the resulting phenotypic character. We will use these branches to track how a change at the scale of a single molecule can determine the difference between metabolic stability and systemic disease. Identifying where an error sits in this hierarchy is the first step towards explaining why one sequence alteration remains silent while another causes the entire protein to fail. Navigating down the site branch, the most granular error is a point mutation, where a single base is incorrectly substituted during the replication cycle. If a purine is replaced by a different purine, or a pyrimidine by a different pyrimidine, the double helix retains much of its geometry. This is categorized as a transition mutation. A transversion mutation involves a cross-category swap, a bulky purine replacing a single-ringed pyrimidine, or vice versa, which creates a more significant structural distortion. Moving to the consequence branch, base substitutions do not always change the final product. Because the genetic code is redundant, many alterations still result in the same amino acid. These are silent mutations. When the substitution does change the specific amino acid produced, it is classified as a missense mutation. If the change creates a premature stop codon, protein synthesis halts immediately. These are nonsense mutations, which typically have a significant inhibitory effect on function. Sickle cell anemia is the definitive clinical example of how a missense mutation alters systemic function. This sequence comparison shows the molecular error in the beta-globin gene, which is a subunit of hemoglobin, the oxygen-carrying protein in red blood cells. The mutation occurs at the 6th amino acid in the polypeptide chain, where the normal glutamic acid is replaced by valine. This single substitution changes the polarity and the folding structure of the entire hemoglobin molecule. Under low oxygen conditions, these altered hemoglobin molecules cause the red blood cells to assume a rigid sickle shape, as seen in these comparative micrographs. These malformed cells clog the capillaries, leading to painful episodes and cumulative organ damage throughout the body. The physical agony of sickle cell anemia is the systemic result of a single microscopic point mutation. While missense mutations swap a single building block, nonsense mutations create a premature stop codon that halts translation before the protein is finished. This results in a truncated, typically non-functional fragment. Beta thalassemia is often the result of a nonsense mutation in the beta-globin chain, which severely compromises oxygen transport to vital organs. Hurler syndrome follows a similar mechanism. A nonsense mutation in the IDUA gene prevents the production of an enzyme needed to break down complex sugars, leading to a fatal buildup in children. A different error is the frameshift mutation, the physical addition or deletion of nucleotides. Because translation reads mRNA in GRI base codons, altering one nucleotide shifts the entire reading frame. Every subsequent amino acid is changed. The result is a total structural collapse of the protein, as every instruction downstream of the error is rendered incorrect. While substitution changes a single component, frameshifts and nonsense mutations disrupt the entire logic of the translational sequence. Navigating the taxonomy to the source branch reveals why these errors occur. Spontaneous mutations arise from internal replication or metabolic anomalies without any outside influence. One major cause is a tautomeric shift, a brief window where a nitrogenous base adopts an unstable structural form. If replication happens in that moment, an incorrect base is inserted and becomes permanent. Spontaneous mutation rates vary. Larger genes are more prone to error due to more opportunities for replication mistakes. Neurofibromatosis type 1 has a high rate, 40 to 100 mutations per million gametes, often appearing with no prior family history. In contrast, induced mutations are those caused by chemical or physical mutagens in the environment that force error rates to spike. The analog 5-bromouracil impersonates thymine so effectively it's incorporated into new DNA. Its chemical instability then forces a permanent change. Across 2 rounds of replication, it shifts bonding preference, mutating an original AT base pair into a GC pair. Whether the cause is the internal instability of a tautomer or the external infiltration of a chemical analog, the DNA replication machinery is perpetually vulnerable to exploitation. The final branches of our taxonomy categorize mutations by their visibility, morphological changes like plant height, and the specific character they affect, such as biochemical function. We distinguish between physical alterations and enzymatic failures, such as phenylketonuria, which is caused by a mutation in a single enzyme. The vast majority of these mutations are damaging, resulting in faulty proteins that cause hereditary diseases or drive the uncontrolled growth of cancer. Yet this constant sequence alteration is the only mechanism that provides the variation necessary for life to evolve. Rare micromutations have allowed insect populations to survive and adapt to the introduction of chemical pesticides in their environment. In agriculture, spontaneous mutations causing polyploidy, extra sets of chromosomes, have produced larger, more resilient crops, including modern citrus fruits. Mapping an error through its site, through its consequence, through its source, and through its character is the only way to explain how life operates at its physical and functional limits. The same molecular instability that triggers systemic pathology also provides the necessary variation for life to adapt. It is the inescapable cost of maintaining a biological system that must replicate billions of times.